Hello everyone, welcome to my channel. Today we are going to talk about bacterial biofilm formation. These are the objectives we are going to discuss in this topic. First one is to know about what does biofilm really means. The second one is to know about the composition of biofilm, and the third is to know about the stage of biofilm production. Before diving into the topic, we should know about what does biofilm really means. Biofilm is an organized community of bacteria enclosed by self-made extrapolymeric substance which are attached to living or non-living surfaces. It is not just a random collection of bacteria, it is a well-organized, social and protected community with specific architecture and water channels across it. There are some key terms which we have to know before diving into the topic. The first one is planktonic bacteria. They are the freely floating bacteria. These are the vulnerable bacteria which can be killed by antibiotics and immune system. The second one is sessile bacteria. Sessile bacteria are the bacteria that are present in biofilm and is protected. Bacteria can interchange between these phases according to the condition. Let's now talk about the composition of biofilm. The first one is extrapolymeric substance. These are also known as slime and is one of the most important composition of biofilm. It consists of protein, lipids, polysaccharides, and nucleic acid. It helps to hold bacteria together, evade immune response, and acts as diffusion barrier for antibiotics. Then the second one is environmental DNA, also known as eDNA. It is released by bacteria, usually through autolysis, and it incorporates into a matrix. eDNA helps to stabilize 3D structure of biofilm and access nutrients during a starvation. Then third one is water channels. Water channels act as a circulatory system across the biofilm. It helps to flow in nutrients and excrete toxic materials. And the last one is bacterial cells. Bacteria of varying metabolic activities are present in outer layer. Of bacteria of very active and metabolically fast are present, whereas in inner layer, slow and dormant bacteria are present. Now, biofilm production. The first stage of biofilm production is irreversible attachment. It is a non-specific and weak attachment. In the beginning, planktonic bacteria approach to the surface. The attachment depends on hydrophobicity, electrostatic force, pH, temperature, nutrition availability, and oxygen availability. Bacteria can easily detach in this phase, so it's easier for the removal. Second stage is irreversible attachment. Bacterial surface appendages anchors to the surface. Major appendages include flagella, fimbriae, lipopolysaccharides, and adhesins. After bacteria irreversibly attach, they undergo change in genetic expression. Third stage is extracellular polymeric substance synthesis and microcolony formation. Bacteria start synthesizing EPS. They also multiply and form small clusters known as microcolonies, which is enclosed in EPS matrix. In this phase, eDNA is also released into the matrix and 3D structure of biofilm begins to form. The fourth stage is maturation. Mature biofilm consists of mushroom-shaped towers, water channels across the biofilm, different zones for different metabolic activities. Quorum sensing is the key mechanism for maturation of biofilm. Quorum sensing. It allows bacteria to act like multicellular organisms to perform a task that would be ineffective for single individual cells. Bacteria release chemical signals called autoinducers. The concentration of autoinducers is proportional to number of bacteria. When bacterial concentration became very large, autoinducers reaches the threshold limit, known as quorum. Reaching threshold results in binding of these autoinducers with the bacterial cell surface receptor, which changes the gene expression of bacteria across the biofilm. At fully mature biofilm, bacteria became immune evasive and antibiotic resistant. The last phase is detachment. Bacteria actively detach from the mature biofilm. There are various reasons for detachment like nutrition depletion, oxygen depletion, accumulation of toxic substance. This detached planktonic bacteria attach to new different surfaces. Now, biofilm and antibiotic resistance. Bacteria in biofilm are 10 to 100 times more resistant than the planktonic bacteria. There are several mechanisms through which antibiotics resistance occur. Some are listed below. EPS slow down and prevent antibiotics to reach inner layers. Then the second one is EPS traps and inactivate antibiotics. Like for example, Beta-lactam is concentrated on the matrix, inactivate the beta-lactam antibiotics. The third one is that the most antibiotics act on metabolically active bacteria, but in context of biofilm, the inner context of biofilm are dormant, resulting in its resistance. The fourth one is altered environment like change in pH, oxygen depletion, nutrient unavailability may reduce efficiency of antibiotics. And the last one and the most important is that the close proximity between the cells cause transfer of resistant gene easier, which results in antibiotic resistance. For detection of biofilm, there is phenotypic and genotypic detection techniques. But in this slide, we are going to talk about phenotypic detection only. There are three methods commonly. First one is congruent agar, then second one is tissue microtiter plate assay, and the third one is scanning electron microscope. First, congruent agar. Congruent agar is a simple qualitative phenotypic method used to detect biofilm forming bacteria based on colony color change in the presence of congruent dye. Its principle is congruent dye binds to polysaccharides in the EPS matrix of the biofilm producing bacteria, producing a visible black color. Non-biofilm producer cannot bind the dye and appear as pink or red color. The second method is tissue microtiter plate assay. Biofilm forming bacteria adhere to polystyrene surface of microtiter plate. The adherent biofilm is stained with crystal violet and measured absorbance at 570 nanometer. Here is the procedure of a tissue microtiter plate assay. First of all, subculture isolates into a nutrient agar and incubate at 37 degrees Celsius for 24 hours. Then, inoculate colony in triplicate soy broth with 1% glucose and incubate at 37 degrees Celsius at 24 hours. After that, culture is diluted with fresh triplicate soy broth in 1 is to 100 ratio. From the diluted solution, 200 microliter sample is placed into a microtiter plate. For negative control, fresh broth is used, while for positive control, organism forming biofilm is used. These microtiter plates are incubated at 37 degrees Celsius for 24 hours. The contents are removed by gentle tapping. Wash 4 times with phosphate buffer saline to remove freely floating bacteria. Then fixed by 2% sodium acetate and stained with 0.1% crystal violet. Excess stain is removed by deionized water and air dried. The absorbance is measured at 570 nanometer using ELISA reader. For interpretation, we should know about the OD and ODC. OD means the absorbance of the test sample, whereas ODC means average OD of negative control plus 3 times SD of negative control. To interpret it, if OD is less than ODC, then it is considered as non-producer of biofilm. If OD is in between ODC and 2 times ODC, then it is considered as weak producer. And if OD is between 2 times ODC and 4 times ODC, then it is considered as moderate producer. If the OD is greater than 4 times absorbance of negative control plus 3 times SD of negative control, then it is considered as strong producer. Thanks a lot for coming this far. If you like this content, please like and subscribe. And if you have any suggestion, suggest in comments. Thank you.